Imagine you're on a luxury expedition cruise ship, traversing the remote, freezing waters of the South Atlantic in April 2026.
Right, so you've got icebergs outside the window, a five-star dining room inside, and you are just, completely, undeniably isolated from the rest of the world.
Yeah, you really are. And then suddenly, a severe respiratory illness breaks out among the passengers. But the thing is, this isn't norovirus or your typical cruise ship bug. It's actually a deadly pathogen that we usually associate with, like sweeping out dusty rural barns and rodent droppings.
Which, makes its appearance in the middle of the ocean both a massive medical mystery and just a complete logistical nightmare.
It really does. And that is the exact scenario that unfolded aboard the MV Hondius earlier this year. So for today's deep dive into the sources, we are pulling from a pretty massive stack of data.
Yeah, we've got urgent CDC Health Alert Network advisories, World Health Organization outbreak reports, and some really fascinating molecular research out of Hungary.
Right. And our goal here is to figure out how a virus that is native to rural environments somehow ended up causing total chaos on a luxury cruise ship.
We're basically going to look at the unique biology of this specific pathogen, how it interacts with our immune systems, and crucially, what this isolated cluster reveals about the current vulnerabilities in our, global health infrastructure.
Exactly. And we are going to skip the panic. We'll avoid all the super dense medical jargon and just focus on the mechanics of what actually happened. So let's ground this in the reality of the incident for you listening.
Yeah.
On April 1st, 2026, the MV Hondius departs from Ushuaia, Argentina.
Ushuaia, right.
Yeah. And we're talking 147 passengers and crew from 23 different countries. They are heading out to these incredibly remote locations like Antarctica, South Georgia, and St. Helena.
So it's a truly international demographic, and they are packed into a highly confined, climate-controlled environment for weeks at a time.
Right, which sets the stage. And then we have case one, the index case. This is an adult male who had been traveling around South America for, I think, 3 months prior to boarding the cruise.
Yeah, 3 months.
Okay, so he developed symptoms on April 6. And then just 5 days later, on April 11, he is dead from rapid respiratory failure.
Wow. And the situation escalated incredibly aggressively from there. By May, the World Health Organization was tracking an outbreak that grew to 9 confirmed cases, 2 suspected cases, and 3 deaths.
So just doing the math on that for a second,
Yeah.
You're looking at a roughly 33.3% case fatality rate.
Yeah, which is staggering.
It really is. It became such a severe threat that 18 American passengers actually had to be actively airlifted by the State Department. And they weren't just taken to a local hospital. They were flown to specialized high containment units in Nebraska and at Emory University in Atlanta.
Which is no small feat. That requires specialized biocontainment transport pods and highly coordinated international logistics. It just highlights how seriously public health officials take this specific family of pathogens.
Okay, well, let's unpack this transmission because this is where the story gets pretty strange. When I think of hantavirus, I think of, someone inhaling aerosolized mouse droppings in an old shed.
Right, the classic rural exposure.
Yeah. So how does a rodent virus spread among passengers on a luxury cruise ship? Did they have a stowaway mouse running through the ventilation system of the Hondius?
Well, that's the most logical assumption, right? If it's a rodent virus, you look for the rodent. But the epidemiological investigation points to a completely different answer. There was likely no mouse on that ship spreading the virus.
Wait, so the transmission was person to person?
Almost certainly. And that detail right there isolates the exact pathogen we're dealing with. The WHO confirmed the culprit is the Andes virus, or ANDV.
Right.
It's native to the southern cone of South America. And what makes the Andes virus entirely unique is that it is the only hantavirus known to science that has been documented to spread from person to person.
Oh, wow. Only the Andes virus does that.
Yeah, exactly.
So case one likely contracted it from a rodent during his 3 months traveling in South America, brought it onto the ship, and then just passed it directly to his close contacts before he died.
That is the prevailing theory right now. Now, we should clarify, it doesn't spread as easily as something like the flu. It requires close, prolonged contact.
Like what kind of contact?
We're talking intimate physical contact, sharing small enclosed spaces for long periods, or, direct exposure to respiratory secretions.
Well, a cruise ship cabin essentially functions as the perfect incubator for those exact conditions.
Unfortunately, yes, it really does. And the Andes virus causes a disease called Hantavirus Pulmonary Syndrome, or HPS. It's a severe lung disease, and the mortality rate generally sits somewhere between 20 and 40%.
Which is terrifyingly high.
It is, especially when you contrast it with the older hantaviruses found in Eurasia.
Right, the old world strain.
Exactly. For instance, the Dubrava-Belgrade virus causes a completely different condition. It's called hemorrhagic fever with renal syndrome, or HFRS.
So renal, meaning it hits the kidneys instead of the lungs.
Right. It primarily targets the kidneys. It is still very serious, but the mortality rate is significantly lower. It's usually between 5 and 15%.
Okay, so the New World hantaviruses tend to hit the lungs, and the Old World hantaviruses hit the kidneys. But how is the Andes virus actually killing people?
Yeah.
Like, what is it doing on a cellular level once it gets inside the lungs?
The biology here is incredibly counterintuitive. The virus itself doesn't actually destroy your lung tissue.
Wait, really?
Yeah, it doesn't burst your host cells open like some other respiratory viruses do. Instead, it specifically targets your endothelial cells. Those are the cells that line the inside of your blood vessels, particularly in the microvasculature, the tiny capillaries in your lungs.
Oh, I see. So it just sets up shop in the lining of the blood vessels.
Yes. But the severe symptoms, that 38% fatality rate we saw in some of the source estimates, it isn't caused directly by the virus breaking those blood vessels.
What is causing it?
It is caused by the host's own immune system overreacting to the infection. Your immune response goes into such overdrive that the inflammation literally pulls those endothelial cells apart slightly.
So it's creating gaps in the blood vessel walls.
Exactly that. It leads to profound vascular permeability. So blood plasma begins leaking out of the capillaries and floods directly into the air sacs of the lungs.
Wow.
Yeah, the medical term for this is non-cardiogenic pulmonary edema. The patient effectively drowns in their own fluids, which leads to rapid respiratory failure and cardiovascular collapse.
Okay, so what does this all mean? It's basically like a burglar who breaks into your house but doesn't actually steal or break anything. Instead, he just trips the alarm system so loudly and chaotically that the police arrive and end up wrecking the house themselves. Your own immune system brings the wrecking ball. The friendly fire is what actually wrecks the house.
That's a perfect way to look at it. It's a devastating mechanism. And because the damage is being driven by the body's own natural defenses, it makes treating Hantavirus Pulmonary Syndrome incredibly complex.
Which brings up a really big question I had while reading through the CDC advisories. If we have a respiratory virus spreading person to person on a ship, and it has a nearly 38% fatality rate, why are health officials explicitly stating the risk to the general public is extremely low?
Yeah.
I mean, why isn't this sparking widespread panic about a new global pandemic?
Well, it comes down to a fundamental principle of virology. It's about understanding the difference between lethality and transmissibility.
Okay.
A highly lethal virus is not inherently a highly transmissible one. The transmission of the Andes virus is just biologically inefficient.
Because it requires that intimate, prolonged contact we talked about earlier.
Partially that, yeah. But the primary limiting factor is actually the timing of the viral load.
What do you mean?
Well, with highly transmissible viruses, people are often highly infectious before they even show symptoms. They walk around the community spreading it blindly.
Like with a cold.
Exactly. But with the Andes virus, the viral load peaks, meaning the point where a person is shedding enough virus to easily infect someone else, well, that only happens when the patient is already severely ill.
Oh, I get it. So by the time they are contagious enough to spread it, they aren't walking through a grocery store. They're already suffering from high fever and severe respiratory distress.
Right. By that point, they are almost certainly bedridden, hospitalized, or just naturally isolated from the general community. The sheer severity of the disease acts as a natural brake on its ability to cause a sweeping pandemic.
That makes perfect biological sense.
Yeah.
But for the person who actually gets infected, that timeline creates a total diagnostic nightmare. I mean, our sources highlight just how hard it is to catch this virus early.
It really is.
Because the initial symptoms, fever, fatigue, deep muscle aches in the thighs, hips, and back, they are completely generic. They just mimic the flu.
And this is the crux of the clinical challenge. By the time doctors start seeing the severe clinical markers, what epidemiologists call the classic five, the patient is already in a state of crisis.
Okay, I saw the classic five mentioned in the outbreak reports. What exactly are doctors looking for there?
So they are looking for a rapid drop in platelets. They also look for hemoconcentration.
Which means the blood is actually getting thicker, right?
Yeah, it gets sludgier because all the liquid plasma is leaking out into the lungs. Then they look for a left shift in white blood cells.
A left shift. What does that signify?
It's a sign the bone marrow is basically pumping out premature immune cells in a panic. And they look for atypical lymphocytes and finally rapid respiratory failure.
Okay, so by the time a doctor actually sees all 5 of those red flags, the patient's lungs are already failing. That friendly fire is well underway. What about testing, though? Can we just swab them early on?
Well, we do have some new technology assisting triage. There's a tool called the MEMED BV Flex test, which recently received FDA breakthrough designation.
Oh, right.
It's incredibly useful because it can tell a doctor whether an infection is bacterial or viral in just 15 minutes.
Which is great for ruling out bacterial sepsis, but it doesn't specifically identify the Andes virus itself. For that, you need a PCR test. And the reports note that the specific PCR tests for the Andes virus are largely labeled research use only.
Yeah, they aren't widely CLIA validated.
CLIA.
Right. CLIA stands for Clinical Laboratory Improvement Amendments. And a lack of validation basically means these tests aren't certified for routine diagnostic use in your standard local hospital lab. They are mostly relegated to specialized research facilities.
Which means critical delays in getting an official diagnosis. And let's say a doctor does correctly diagnose the Andes virus on, like, day 3. What can they actually prescribe?
Our sources point to a massive antiviral gap here. There is currently no FDA-approved treatment for Hantavirus Pulmonary Syndrome.
None. The standard protocol is entirely supportive care. If the lungs fail, doctors rely on interventions like ECMO.
Extracorporeal membrane oxygenation. Exactly. It's a machine that pumps the patient's blood outside their body, oxygenates it, and pumps it back in, completely bypassing the lungs to give them time to heal.
But that's just buying time. What about actual antiviral drugs? Have we made any progress there?
Doctors have experimented with off-label antivirals. Ribavirin, for example, has shown some limited success treating the kidney-targeting hantaviruses if it's administered very early. But it remains unproven for lung-targeting infections like the Andes virus.
And what about that other one, favipiravir?
Unfortunately, favipiravir is still stuck in the preclinical testing stages.
Wow. I read the insights from epidemiologist Dr. Celine Gander in our stack, and she pointed out that there actually were highly promising fully human monoclonal antibodies developed for this.
Yes.
They were created at the Albert Einstein College of Medicine and USM Ride, and they successfully protected animals from the Andes virus. But they were shelved because the NIH funding expired in 2024. The research literally just stopped.
It is a stark reminder that scientific progress is often entirely tethered to funding cycles. And Dr. Gaunder's reporting goes further, pointing to systemic vulnerabilities across the broader public health infrastructure that really complicated this specific response.
And just to be super clear to you listening, our sources are strictly outlining the logistical realities here. We are making political arguments. We are simply looking at the state of the infrastructure as reported by the epidemiologists managing the outbreak.
Exactly. The logistical reality is that the U.S. public health infrastructure was operating under considerable strain during this event. The CDC director position had been vacant for 8 months. The U.S. had withdrawn from the WHO. Funding cuts to BARDA, which is the agency responsible for developing biomedical countermeasures, had completely stalled new vaccine contracts.
Right. And Dr. Gounder also highlighted secondary effects from cuts to PEPFAR, the global HIV program. Wait, what does HIV funding have to do with a hantavirus outbreak on a cruise ship?
I know, it seems unrelated, but it comes down to shared infrastructure. PEPFAR funding didn't just buy HIV medicine. It actually helped build global genomic sequencing networks across various continents.
Oh, I see.
Yeah, so when those funds are cut, the sequencing labs lose capacity. Dr. Gounder noted that South Africa was able to quickly sequence the virus from the cruise ship specifically because of those past infrastructure investments, which are now actively being degraded.
Man, that lack of funding is terrifying when you realize how hard this virus is to spot, even with a fully funded lab. Like, look at this recent 2018 to 2025 MDPI study out of Hungary. They were studying the Dubrava-Belgrade hantavirus.
Right, the old world strain that causes the kidney issues.
Right. The researchers found that detecting the viral RNA is highly dependent on exactly how you process the blood sample. They discovered that sometimes the viral RNA was only detectable in whole blood. If they tested the blood serum or the urine, it came back negative. Why does that matter so much?
It matters because of standard lab protocols. When a hospital lab draws blood, they typically spin it down in a centrifuge to separate the clear serum from the red blood cells and platelets.
Okay.
And they usually just test the serum. But hantaviruses actually bind directly to red blood cells and platelets.
So by spinning the blood down and removing those cells, labs were literally throwing the virus in the trash before running the test.
Yes.
Yeah.
A test could come back completely negative even if the patient is deeply infected, simply because the virus was hiding in the cellular matrix they just discarded.
That is wild.
It demonstrates how incredibly nuanced the diagnostic protocols must be, and why having well-funded specialized reference laboratories is an absolute necessity.
Here's where it gets really interesting for you as a learner. The science itself works. We know how to sequence the virus. We deeply understand the friendly fire biology of how it causes blood vessels to leak. But the logistics, the funding for therapeutics, the global sequencing networks, the basic availability of a validated test in a standard hospital, that is where the armor's cracking.
Which forces us to look at the environmental drivers, too. If our infrastructure is vulnerable, we need to understand why these spillovers are happening now and what innovations are racing to catch up.
Let's look at the root cause. Why was there a massive surge of over 200 hantavirus cases in the Americas throughout 2025 and early 2026? It actually comes down to something called a masting event.
Yeah, a masting event is a massive ecological cascade triggered by climate conditions. In this case, unusually heavy, prolonged rainfall in the southern cone caused native plants to produce a synchronized massive surplus of seeds.
Which creates an unlimited food supply for the local wildlife.
Exactly, which triggers an explosive population boom for the long-tailed pygmy rice rat.
The primary natural reservoir for the Andes virus.
Precisely.
So suddenly you have millions of extra infected rats in the ecosystem. Add in human encroachment, people building homes and farms deeper into these habitats, and severe biodiversity loss. Because there are fewer natural predators like owls or foxes to eat the rats, the human-rodent interaction just skyrockets. And it is not just a South American issue, by the way. Researchers at the University of New Mexico found that hantavirus is carried by over 30 different small mammal species in the U.S. Southwest alone. It's not just the famous deer mouse anymore.
The ecological net is undeniably widening. But science is responding with some incredible innovations. One of the most critical breakthroughs highlighted in our sources comes from the University of Bath regarding vaccine stabilization.
Oh yeah, I saw that in the notes. A process called insilication. How does that actually solve the distribution problem?
It solves the cold chain problem. Most vaccines require strict refrigeration from the factory all the way to a remote village.
Right.
If the temperature fluctuates, the vaccine proteins unfold and degrade. It's estimated that this causes up to 50% of vaccines to be wasted globally. Insilication uses microscopic silica cages to physically encase the vaccine proteins.
Wow, so it essentially shrink wraps the vaccine at a molecular level.
Precisely. It locks the proteins in place so they cannot unfold, meaning the vaccine doesn't need refrigeration at all.
Right.
It can sit at room temperature indefinitely.
That's huge.
It is a game changer for rapid outbreak response in remote areas.
Contrast that with another candidate in the notes, the DNA-based PWRG/ANDM vaccine.
Right.
While it successfully produces antibodies, it currently requires up to 4 separate doses. Logistically, you just cannot ask people fleeing an active outbreak zone to come back to a clinic 4 times over 6 months. You need a stable, single-dose solution that can survive in a backpack without ice.
Exactly. And while researchers innovate on vaccines, the medical community is fiercely debating how to protect healthcare workers on the front lines right now.
Right, the airborne debate published in the British Medical Journal.
Yes. Following the MV Hondius cluster, experts are arguing that we need to upgrade our baseline assumptions. Historically, because hantavirus transmission is so rare, hospitals relied on standard droplet precautions, basically standard surgical masks.
Okay.
But epidemiologists are arguing that the cruise ship outbreak proves we must assume a baseline airborne risk for the Andes virus.
Meaning, they want to mandate N95 respirators and specialized negative pressure ventilation universally for any suspected case. If the virus can spread efficiently through shared cabin air on a ship, healthcare workers in enclosed hospital rooms obviously need the highest level of respiratory protection, without exception.
It is a completely necessary evolution of our safety protocols, based directly on the realities observed during this outbreak.
Okay, well, let's bring all these threads together. We've traced a microscopic threat starting from heavy climate-driven rains in South America. Those rains fed a massive population boom of rice rats. One of those infected rats crossed paths with a traveler, who then boarded a luxury cruise ship.
And the virus exploited the close quarters of the MV Hondius, spreading person to person, triggering that devastating immune system-friendly fire inside the blood vessels of the passenger's lungs.
And it ultimately ended with international repatriations and patients fighting for their lives in high containment units in the U.S. It's a story that highlights massive gaps in our diagnostic availability, therapeutic funding, and global monitoring networks. It is a highly lethal pathogen that is, thankfully, biologically limited in how efficiently it can spread.
It's a powerful reminder that human health is intrinsically linked to our environment. A shift in rainfall patterns thousands of miles away can directly result in an emergency biocontainment airlift over the Atlantic Ocean.
Which leaves us with a final, lingering question to mull over as we close out this deep dive. Consider this, you're listening. The Andes virus is biologically restricted. It requires intimate contact, and its viral load only peaks when the patient is already bedridden and incapacitated. But what happens if the next zoonotic virus that jumps from an animal to humans possesses the 38% fatality rate of the Andes virus, combined with the silent airborne transmissibility of the common cold?
I mean, it is the ultimate worst-case scenario for global public health.
If our global health infrastructure, from the availability of a simple diagnostic test at your local hospital to the international genomic sequencing networks, is actively being degraded, are we simply relying on the biological limitations of these viruses to save us, rather than our own proactive preparedness? It's like leaving your front door wide open and just hoping the burglar decides to sleep in.